Hi and Assalamualaikum warahmatullahi wabarakatuh. My name is Nadia Nur Alisa binti Abdul Ghani, and today we are going to learn on how to generate a 3D model protein using Swiss model. Firstly, get the FASTA protein sequence from UniProt. Search in the UniProt for the protein we desired, scroll down to the sequence section and click FASTA to copy the full amino acid sequence. Next, start modeling the protein on Swiss Model website. Paste your FASTA sequence into the box and click Build Model. Then, the Swiss Model will search for a template structure from known 3D structures like PDB and trees, align your sequence with the template, and build a 3D homology model. For example, here are the 3 proteins that were generated from earlier steps. The first one is ribonuclease H2 subunit A. This protein can be found in eukaryotic organisms including humans and mice. The UniProt ID for this protein is 075792 with the length of 299 amino acids. The ribonuclease H2 subunit A forms a heterotrimeric complex with subunit B and subunit C. Its C-terminal region is an eukaryotic adaptation essential for binding the accessory subunits, which facilitate the formation of a stable and enzymatically active complex. The function of this protein is to cut out bits of RNA that accidentally get stuck in the DNA to keep our DNA clean and working properly, especially during DNA replication, where it helps in fixing the Okazaki fragments and clear harmful RNA-DNA mixes. The second protein that has been generated is the SIG fibroins. SIG fibroin is a structural protein primarily produced by the silkworm like Bombyx mori and is the main component of SIG fibers. Other than that, SIG fibroins also can be found in Haplombia soleri. The UniProt ID for this protein is F6K0S0. It consists of 2 main polypeptide chains, which is heavy chain and light chain. In secondary structures, SIG fibroins exhibit multiple crystalline forms, such as SILK1, SILK2, and SILK3. SIG fibroins are important in building SIG fibers because it can give the silk strength and flexibility to help silkworms make a strong and durable cocoons. Next, because of it is safe for the body and breaks down naturally, it is used in tissue repair, drug delivery, and growing cells in the labs. Thanks to its strength and lightness, SIG fibroins is also used to make eco-friendly plastics, optical devices, and strong blended materials. Lastly, the third protein is bacteriorhodopsin. Bacteriorhodopsin can be found in the purple membrane of the archeon halobacterium selenarum. This protein-rich membrane region is characterized by its vivid color attributed to the presence of bacteriorhodopsin and associated pigments. The UniProt ID for bacteriorhodopsin is P02945, which consists of 248 amino acids featuring 7 transmembrane alpha helices, which forms a compact structure that spans the lipid bilayer. These proteins are important in proton pumping, energy conversion, and also photocycle dynamics. Despite the differences that have shown between these 3 proteins in terms of their characteristics and functions, I have also summarized the structural similarities of these 3 proteins. To sum up, all of it are made of amino acids and fall into 3D structures. The 3 proteins are common use of alpha helices and also beta sheets. All of it rely on hydrogen bond and hydrophobic interactions for their stability. Last but not least, I will explain to you what will happen when these proteins are denatured. When ribonucleus H2 subunit A denatured, it can lose its shapes and functions when heated, exposed to harsh chemicals, or affected by mutations. Mutations, especially at parts where it connects with other subunits, can stop it from working properly. If it doesn't work, harmful RNA-DNA hybrids speed up, which may cause immune disorder like AGS. This disorder will make the body wrongly think there's an infection and attacks itself in this condition. Next, the denaturation of seed fibron can break its strong beta sheet structure, making it weaker and less useful. It can cause by heat, pH changes, or solvents like methanol. Sometimes, controlled denaturation is helpful. In example, to turn into hydrogels or films for medical or material uses. Lastly, the denaturation of bacteriorhodopsin can break key bonds that keep its shapes, especially the parts that span the membrane. This can cause by heat, extreme pH, or detergents, which can break its multi-unit structure. When denatured, it can't move protons or use light energy, which lose its main function. That's all from me. Thank you.